So in this video, we're going to be talking about facilitated and active transport. And I've actually combined all the videos into one today, so I apologize for this podcast. It will be a little bit longer, but you can stop and take a break at any point. So proteins alter membrane structure and function. Okay, so we talked a lot in the last video or in the last class about how membranes are fluid and how they're semi-permeable and how ions move through the membranes. But really proteins in the membrane contribute quite a bit to their function. And proteins can be found in the membrane, so these can be integral membrane proteins, right? Or these can also be called transmembrane proteins, or they can be found near the membrane, and these are called peripheral membrane proteins. And so proteins inserted into the membrane, like lipids, can be amphipathic. So they can have nonpolar regions and polar regions. And they can fold into shapes like any other protein, okay? So you remember when we talked about chaperones, right? Chaperones can kind of protect those nonpolar parts of the protein until those proteins are delivered to the membrane. And then those nonpolar regions will be inserted into the hydrophobic regions of the plasma membrane. Okay, so this is where all those R groups that are just the carbons and hydrogens, right, are found in this part of the protein that's going to be inserted into the plasma membrane. And then up at the top here, right, these amphipathic proteins can have polar R groups right at the top of the protein and at the bottom where it interfaces with the aqueous environment. And then also within this protein here are regions, much like an active site, that interact with ions or other things that might travel through this protein. And we'll talk about that a little bit more. And so this model to describe how proteins are found in the membrane is called the fluid mosaic model. And it suggests that some proteins are inserted into the lipid bilayer, thus making the membrane fluid and a dynamic mosaic of phospholipids and proteins. Okay, so this is a depiction of this phospholipid bilayer, right? It's dynamic, there's proteins in it. And this replaced what was called the sandwich model, where it proposed that the proteins were really just interacting with the membrane but not found in the membrane. And so this fluid mosaic model is the favored model for the description of the plasma membrane of cells. And they have experimental evidence to support this model. And so this was a cool technique where they essentially fractured a cell. So they freezed it and fractured it, and they separated it into two parts, right? So they were able to kind of plop off one part of the membrane and leave the kind of middle intact. And then they looked at microscopy, right? So here's the membrane interior and the membrane exterior, right? And so what they interpreted, right, were these, they essentially saw these divots here and then these mounds there. So what they interpreted was that here is a membrane protein that's sticking into the membrane, right? And then there's the divot where it belonged. Okay, so that this told them that there were proteins in the membrane. And we study membrane proteins a lot in the laboratory. I studied one in graduate school. And the way you get them out of membranes is by using detergents. So if you think of dish detergents. And what these detergents are small, essentially amphipathic molecules that form micelles, okay? And you put this on to a cell, right, that's got a plasma membrane. And these lipids then kind of interact with the hydrophobic stretches and begin to separate the membrane, and they interact with hydrophobic parts of the protein too. And so when you incubate a cell with detergent, you can then separate off the lipids and you can isolate your membrane proteins, okay? And when you isolate a membrane protein, right, and it's intact here, you can ask questions about it, and you can that's helped us identify over the years a variety of different proteins, including proteins that are important for membrane transport. And we're going to talk about those today. And those are channels, carriers, and pumps. And we're going to talk about each one of those in detail. So here's the list of all the different kinds of transport that occurs in the cell. And we're going to talk about each one of these one by one. And we've already talked about simple diffusion. So that's molecules kind of diffusing through this plasma membrane There's lipid plasma membrane here. Okay, so we've talked about that. And now we're going to talk about facilitated diffusion, right? Passive facilitated diffusion, and then active transport, and the different types of active transport. So facilitated diffusion occurs either through a channel or a carrier protein, and it allows only a particular type of ion or small molecule to pass through it. So because of the R groups in the protein, it only has specificity for a single kind of molecule to move through it. And molecules in this group of facilitated diffusion are always going to move from areas of high solute concentration to areas of low solute concentration. And this is going to happen spontaneously because it increases entropy. Okay, so let's talk about our first example of a channel protein called aquaporin. And aquaporin is a water channel, okay? So what this means is that this protein has a pore, and it's specific only for water, and water will move from areas of high concentration to low concentration through the channel. Okay, so that's an example of a channel protein called aquaporin. Oh, this is different than the movement of water by osmosis, right? Because osmosis, it's going just through the membrane, and in aquaporin, it's going through a specific channel. There are also ion channels, right? So here's our ion channel here. It's specific only for one type of ion, and so the ions will move from high concentration to low concentration. And this is really the only way for an ion to get through the membrane. So ion channels are really important. Electrochemical gradients occur when ions build up on one side of the membrane, okay? So here in this membrane, there's a whole bunch of sodium ions, essentially creating a positive electrical charge or an electrochemical gradient on the outside. And so sodium ions will move down their electrochemical gradient, right, into this area that has this negative charge here because it's mostly chloride ions and not a lot of sodium. Okay, so the ions will move down their electrochemical gradient, right, to kind of disperse the charges. And here's an example of what that would look like. And so here we're just measuring current. And so this is measuring an electrochemical gradient. And so if we stuck an electric probe in this water here, we would really see no electrochemical gradient, right? This is just pure water. There's no concentration of ions. And if we stuck it in Gatorade that has a whole bunch of electrolytes, we stuck our probe in, right, we'd get a reading, boop, 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 a current, right? Because there are electrolytes, okay? And so what I'm really saying when I say electrochemical gradient is that there's a concentration of ions, or a concentration, essentially, think of Gatorade, of electrolytes, right? We can detect a charge. Now I want to talk about a disease, and that's cystic fibrosis. And cystic fibrosis occurs by mutations in a gene called CFTR, okay? And CFTR is known to be a chloride ion transporter. And so if you don't have CFTR in a cell, you don't get chloride ion transported, okay? So there's no chloride detected, right? There's no current. But if you stick that chloride transporter into the cell, right, all of a sudden you get that current, you get that reading from that electrical probe. So without the channel, ions are not pumped in and out of the cell, okay, in this disease. So if you have a mutation in CFTR that doesn't give you a functional CFTR protein, you don't get ions pumped in and out of your cell. And this actually results in a mucus buildup. So they have a really hard time breathing. They've got a lot of mucus in their lungs. And that mucus also creates a really nice environment for a lot of bacteria to grow. And so they have a lot of bacterial pneumonia problems from bacteria that accumulate in that mucus. And so ion transporters are very important. There are also what's known as voltage-gated ion channels, okay? And channels, right, are just proteins that ions can diffuse down their concentration gradient, so they can go through the protein. But sometimes they can be opened and closed, okay? And these voltage-gated channels, that was just an example, there, can open or close when they detect an electrochemical gradient. Okay, so let's say that we have I'm going to go back real quick. Let's say we have a positive charge on the outside of cells. Okay, so we have this electrochemical gradient here with all these positively charged ions on the outside of cells. And when this situation occurs, this voltage-gated channel, potassium channel, for example, is closed. Right? There's already too many positive charges out here, so this channel is just going to be closed. So then the ion can't pass through because the channel is closed. But let's say something happens on the outside of the cell where now there's all these negatively charged ions on the outside. Well, then the channel opens and potassium can flow down its electrochemical gradient outside the cell. So that's an example of a voltage-gated channel, a channel that can open and close in response to an electrochemical gradient. The next type of facilitated diffusion transport I want to talk about are carrier proteins. And carrier proteins are like channels, so molecules are going to move down them, but down their concentration gradient, but they're actually going to change conformation to allow that molecule to move through the membrane. And so GLUT1 is an example of this transporter. So here we have a molecule of glucose, and I'm going to go back. Before glucose is bound, GLUT1 is in this conformation here, and then glucose binds. And when glucose binds, that changes the conformation of GLUT1 and allows glucose to exit the cell. Okay, so the difference between channel and carrier proteins is that carrier proteins change their conformation when they bind their solute. Okay, but everything's still moving down their concentration gradient. So now we're going to switch gears. So if you need to take a break in the video, it's a good time. But we're going to talk about active transport. And so active transport is basically saying that we now want to move molecules against their concentration gradient. So we're going to take areas that have low solute concentration and move those molecules to areas of high solute concentration. And this requires energy, okay, because we're decreasing disorder, right? We're concentrating chemicals in a certain space. Okay, so molecules are pumped against their concentration gradient, and this needs energy, and it's usually in the form of ATP. So the classic example of active transport is the sodium-potassium ATPase. Okay, and you're going to see this again and again and again throughout your education and in exams and wherever you go afterwards. People will be using this example for active transport. And so this is how the sodium-potassium ATPase works. So you have this protein embedded in the membrane here, and it's what this protein has is 3 sites for sodium to bind and 2 sites for potassium to bind. And so what's going to happen is sodium that's inside the cell is going to bind to the sodium-potassium ATPase, and then ATP is going to come along and it's going to transfer a phosphate group to the sodium-potassium ATPase, right? And then you're left with ADP, and the ADP will diffuse away. When this phosphate binds to the sodium-potassium ATPase, the sodium-potassium ATPase is able to change conformation. Okay, so now it's open to the outside in its conformation, and in this conformation, it loses its affinity for the sodium ions. So the sodium ions are now pumped against their concentration gradient. So there's a lot of positive charges. And then potassium travels down its concentration gradient, so it goes back into the ATPase. Okay, so there's sodium in the potassium in the ATPase. The phosphate then dissociates from the sodium-potassium ATPase, so it returns to its conformation, right? That's original conformation, where it loses its affinity for potassium, right? Because it had affinity for potassium in the other conformation, it doesn't in this one. And then potassium then diffuses into the cell. And so what we've done now is we've set up an electrochemical gradient. So now there's more positive charges. There's this concentrated sodium outside of the cell versus inside of the cell. right? Which has only got, you moved 3 positively charged ions out for only 2 positively charged in. So if you look at membrane potential across the cell membrane, you'll see that this is often represented by a bunch of plus signs, right? So this is usually slightly positively charged on the outside because of the function of the sodium-potassium ATPase. And our cells spend a ton of energy generating this electrochemical gradient. So a lot of our cells spend the ATP making this gradient. So again, this is the picture from your book. We have 3 sodium ions going in, we have ATP binding, changing the conformation, then sodium loses its affinity, sodium goes out, 2 potassium that now have the affinity for this conformation come in, right? They bind, the phosphate dissociates and it changes back to shape, and then the potassium are spit out. So this is how the sodium-potassium ATPase works. Okay, so that's active transport. But now we have this sodium out here. We used energy to get it out here. What are we going to do with it? Why did we put it out there? And one of the ways in which we can use it is to use it for secondary active transport. Okay. Okay, so secondary active transport, it uses energy indirectly. Okay, so it's going to use the ions pumped against their gradient, so these sodium ions, meaning that it's going to use the established electrochemical gradient. Okay, so it's not going to directly use ATP, but it's going to use something that ATP was used to do. Okay, so the classic example of a secondary active transporter is the sodium glucose symporter. So symporter just means that both molecules are going to move in the same direction. Okay, so here we have our sodium that we pumped out of our cell, and we've got a molecule of glucose. Okay, now we would like to get as much glucose into our cell as possible because we're going to use that glucose to make ATP. Okay. So we want to be able to pump glucose against its concentration gradient. And we're going to kind of use the sodium that we pumped out to do this. Okay, so here we have our sodium glucose symporter. And glucose is going to bind and sodium is going to bind. Okay, now sodium is now moving down its concentration gradient. And this glucose, we're saying, is going to move up its concentration gradient. So let's pretend we already have a whole bunch of glucose in our cell, more than we possibly need. It doesn't matter, we still want more. Now once these two bind, here they're bound, they're able to now move through the symporter. Okay, so they're both moving in the same direction. So we were able to now use the sodium moving down its concentration gradient to move glucose up its concentration gradient. And this is called secondary active transport. Okay, so it's using the energy indirectly. So just to summarize the different kinds of transport we talked about, there's passive transport, which is diffusion, facilitated diffusion through channels, and facilitated diffusion through carriers. Those are the ones that change shape. There's active transport, there's primary active transport, in which that energy from ATP is used to pump ions or solutes against their concentration gradient, and secondary active transport, where you use that electrochemical gradient to move something else up it or against its concentration gradient. So that's it for today's video. I'm sorry for it going a little long, and I look forward to seeing everyone in class. Thanks. Bye.